Welcome back for more AP Biology. Today we're going to be dealing with linkage and the issues associated with linkage. So what happens when chromosomes don't separate? Which is not a linkage issue, but it kind of is. There's quite a few things that you'll get tested on, references to the book. So if we look at where we've been so far, I've told this story before. We started off dealing with statistics, which then led to us talking about what we could see, which is ecology. From there, we deal with natural selection. Natural selection begs the question of how does inheritance work? So we had to deal with inheritance. Inheritance then can be merged with natural selection. We get what we call population genetics or the modern synthesis. From there, we've gone as far as we could see, so we need to then venture into the world of cells. From cells, we could deal with how cells deal with movements of substances and how cells can turn out to reproduce. The catch is, that's all we can really talk about with cells. To do anything else, we need to speak in terms of chemistry. So we're going to start talking chemistry. In particular, very small molecules that we call the macromolecules. We've dealt with those before, but we're going to deal with them in what they do. Mitosis and meiosis, which are both cellular reproduction processes, have very similar aspects to them, yet they come up with very different outcomes. We can use meiosis to explain part of Mendel's laws of inheritance. The catch is they don't always work, and it's because of those chromosomes that Mendel's laws don't always work. We call those failures linkage groups, or when you turn out to have dependent assortment, it's not an independent assortment, meaning the genes are somehow stuck together. So how could we see this? We could be given data that look like this. So when I look, this is dealing with Drosophila, which are fruit flies. You could deal with the body types, which are gray or black, but you could also deal with what their wings look like. So are they gray, or pardon me, are they normal looking, or are they all shriveled up, what we call vestigial? If I were to sit there, take two flies that are homozygous, so true breeding, cross them, I get a heterozygote. If I were to then sit there and say, oh, well, let's do a test cross or whatever. So I'm going to cross them with one that is homozygous recessive. What I would expect to see is even distributions. That's not what I got here. I actually have it, so it's predominantly gray vestigial or normal black, and then I have these two freak ones, or normal gray and black vestigial. I didn't get that. I expected this, but I got something else. The question here then becomes, well, does that mean Mendel was wrong? And we could test for that. It's called a chi-square. So you do the chi-square and you get 1500. I don't know what, so degrees of freedom would be 3. I don't know what p-value you want to pick, but I'm pretty sure that a chi-square to 1500 is going to be greater than the critical value that you picked, which means Mendel's wrong. And chromosomes show it. What we can do with that phenomenon, the fact that these genes are linked, is we can do a little bit of math to figure out what we would call a recombination frequency. And that recombination frequency, which is how often genes trade spots, meaning they don't match the pattern, meaning if I were to look at the gray vestigial, that seems to travel together. Normal black seems, or black and normal seem to travel together, even though it says back, not black. It's not my picture. I stole the picture. They seem to be traveling together, but it seems to be weird to get a gray normal or a black vestigial. That seems to be a little weird. So what I could do is I could calculate out, well, how often does that happen? When you calculate that out, we get what we would call recombination frequency, and two individuals, so Thomas Hunt Morgan and one of his postdoc students, whose first name I think was Alfred, I can't remember, but it's Stervant. It's the last name that I look at, and I've known of him for 20 years, and it's always, I don't want to say this name. But anyway, what they did was they took the information. They realized, oh, I can make a map out of this, which is what this picture is showing you. We can actually take recombination frequencies, RF, and string things together. There becomes a very odd pattern, however, that we need to realize. And that is, if I look at how these things add up. So if I look at B, so I know C and D are saying stuff, but if I just look at B, the recombination frequency between a Y and a W, whatever those traits are, is 1.1, meaning 1.1% of the time there's a recombination. Between the W and the M, the recombination was 32.8% of the time, which means how often should it happen between the Y and the M? 1.1 plus 32.8, which is, what, 43.9? 43.9% of the time, I should see a recombination frequency between Y and M. And the answer turns out to be, no, it's actually 34.3. Your response is, wait, what? That doesn't make sense. And it's because we're dealing with probabilities, not actual percentages. So recombination frequency is actually dealing with a probability. It's not a probability and a frequency, which is a statistical phenomenon, not just a, well, here's what the percent is, and the percent is always the percent. Recombination does not add up, which means we don't turn out to use the unit of percent. We use different units that we'll talk about later. The result is I can actually start to put traits in locations. And what I get is something that looks like this. This turns out to be a chromosome map for a handful of traits dealing with Drosophila. They turn out to have 4 chromosomes. They have 3 autosomal chromosomes, so ones that have nothing to do with sex, and they have 1 pair of sex chromosomes. They have an X and a Y. And the result is everything that you see here. That's one thing that this linkage can tell us about. But linkage also could be the 2 chromosomes or the chromatids stuck together. And what can we learn from that? The first thing that we could learn is there needs to be some type of mechanisms that let the chromosomes separate, the homologous pairs of chromosomes and the chromatids separate. What we notice is that center part of the chromosome, what we call the centrosome, it is actually surrounded by a whole bunch of proteins called the kinetochore. The kinetochore, when aligned up perfectly, is capable of separating. When they don't line up, they don't separate, or at least they're not supposed to separate. And clearly there's something going on here that tells the cell that the kinetochores are free to separate or they're not free to separate. There's some type of communication going on. Whatever it is, there's some type of communication. We also know that they don't immediately separate. They actually move and they wiggle and jiggle until they're in the middle, and then we'll see them separate, which means they have to know that they're in the middle. If we look closer, what we notice there are proteins, microtubules, what we call the mitotic or the meiotic spindle, will stretch out from the origins of microtubules. They're called centrosomes, with little things called centrioles. So what you can do is we can attach those microtubules to the kinetochores, and what they can do is attach, wiggle and jiggle till they're in the middle, and pull apart. Something is telling it it's in the middle, but we now know how we can get them stuck in the middle. The catch is I need to split the cell in half, so I can obviously reel these 2 sides in, but that's only going to go so far. So if you look at this picture, there are other sets of microtubules that kind of stretch out and kind of kick back from each other. And the result is I can pull the chromosomes apart and then push them apart. We have a push and we have a pull going on. How does it know to do this? There must be some method of communication, which means we're going to be running into very shortly, how do these things know to do this? So what if it doesn't do what it's supposed to do? The result is you have too many or too few chromosomes. That phenomenon is called a non-disjunction. And depending on the result, we can actually tell you what went wrong. Additionally, non-disjunction is not exclusive to meiosis. It could happen during mitosis, because after all, we have to still separate the sister chromatids. which means we run the risk of that failing too. What we also have going on outside of non-disjunction is we have that crossing over thing. And crossing over could go wrong as well. Depending on how the crossing over works, we could end up deleting sections of genes or of chromosomes. We could duplicate parts. Meaning we could actually slam them together and we could have double parts on a chromosome. Because if you have a deletion, you're going to have a duplication somewhere else. We can, have crossing over on the same chromosome and switch parts. And now we put things in a different order, which might be bad. Or we could have crossing over between two totally different chromosomes. And the result of that is very strange. In particular, it is so strange that there is a woman by the name of Dr. Rowley. She died a couple of years ago. She examined the genetic composition, the karyotypes of individuals who happen to have something that we call chronic myeloid leukemia, which has a whole bunch of issues that go wrong with it. It's a form of cancer. It's a white blood cell cancer. And it's not particularly fun. But it turns out that this phenomenon, this particular type of cancer, shows up due to a translocation, so crossing over between two different chromosomes, non-homologous pairs, of a 9 and a 22. It makes this weird fragment of a chromosome that is associated with people who happen to have chronic myeloid leukemia. What makes this strange is chronic myeloid leukemia was first characterized in Philadelphia, well before Dr. Rowley did her work. When she did her work about people with chronic myeloid leukemia, she started to notice this pattern that this little itty-bitty chromosome was showing up, and it was thus dubbed the Philadelphia chromosome because of the fact that chronic myeloid leukemia was first characterized in Philadelphia. It has nothing to do with the city. It's just an association.